we have Dr. P Talwar और मैं थोड़ा सा introduction आपको देना चाहूँगा उनका. Dr. P Talwar is a biomedical researcher working in the area of vaccines and immune contraceptions. He is Director Research at the Dr. Talwar Foundation, a scientific and industrial research organization recognized by DSIR, Government of India. He was professor and head biochemistry, All India Institute of Medical Sciences, New Delhi, 1965 to 1983. He was elected as the president of the International Society of Immunology of Reproduction and was the first president of the Federation of Immunological Societies of Asia Oceania. Professor Talwar was conferred Padma Bhushan by President of India and Officer de la Légion, the honor by President of France. He has published over 600 publications in reputed journals. He is editor and author of 10 books and monographs. Can we have the huge round of applause? Sir, you are welcome here once again. Thank you. And today's topic of the presentation is Immunotherapeutic Approaches for Advanced Stage Difficult to Treat Cancer.
Thank you very much. Have my slides gone to the place? Well, I'm going to speak on, this is the menu there, where we have a basic research carried out on a problem which goes to a lead and then eventually to make it a product which can go to the public. This is the sort of channel we follow. Obviously you have to do toxicology, then the approvals of the drug controller, of ethical committees at various steps, and then to carry out phase 1, phase 2, phase 3 trials. Then it gets approved by the DCDI and you can give it to industry to make it available to public. So this is the route that we follow, and what I'm going to tell you is the route that we followed for some cancers. Immunotherapy of cancers is our recent interest in recent years. May I have the next slide, please? Next. Now, HCD cancers is unexpected. HCD is an index of pregnancy. It's a diagnostic test for pregnancy. Whenever the human chorionic gonadotropin is detected either in blood or urine, that is the way that we had always associated HCD with. It's with pregnancy we have associated it. But you see, what is noticed is that a whole category of cancers which are at the advanced stage and they are drugs resistant and they are almost, the person suffering from it is almost gone, literally. These cancers also express ectopically. You least expect a cancer to express HCD. It's related to pregnancy. But there you have a whole group of cancers which start expressing HCD ectopically. And that is what I'm going to speak about today. Next slide. These are many people in the world who have worked on it. And here is lung carcinomas, pancreatic carcinomas, bladder carcinomas, colorectal cancers, gonad tumors, lymphomas, lymphoblastic leukemias. In all these fields, there have been publications of ectopic expression of HCD, the pregnancy hormone. Next slide. And what is further noted is that in each case, everywhere in the world, you see those tumors which are expressing HCD have very little chance to get cured and they all die in a short time, short or longer time, time varies. Next slide. Now you see HCD, what is interesting about it is that it is also a very good target for immunological approach. Why? Because HCD is not made by any normal organ of the body. Nowhere. It is made, and it only comes into being when pregnancy occurs, when sort of fertilization leading to conception takes place. And therefore, as HCG is not made by any organ, any normal organ of the female, it says ion of the male, of course. Male never makes HCG unless he is suffering from one of these cancers. So therefore, if we adopt or try to seek an immunological approach, it will be highly safe because normal human tissues do not react with anti-HCD antibodies. Next slide. Next slide, please. And here it is an indication, we have not only written, but we have even tested that in all the kidneys, 5 different organs of the body have been tested, and they don't react with anti-HCD antibodies, obviously because HCD is not made by any other organ of the body except when the conception takes place. Next slide. We made a monoclonal antibody which we have named as PIPP. This antibody was made during my Nehru Fellowship days. These 2 years I was a Nehru Fellow. And what is interesting about Nehru Fellowship is that normally what you do belongs to the institution where you work. But whatever you do during the Nehru Fellowship days belongs to you. These are the sort of privileges of the Nehru Fellowship, 2 years fellowship that is given to those who deserve it. And so you see, during that time, we made this monoclonal antibody, which is named as PIPP, Pratirakshak Immunobiological Products and Processes, is a company I had to form because this very antibody has been licensed to Carter Vallis, USA, who sent us actually such a good antibody of high affinity and specificity. So they wanted to license it, to take it for their own commercial making as diagnostic kits. And they sent us tickets to come to USA along with a lawyer who can read what the agreement will be. And I did go there. We made an agreement and they gave me $60,000 after signing that agreement. I thought, well, that's sufficient. You see, normally in India, we hardly, the companies take your product, give you a little bit in the beginning, and then thereafter, it's rarely that they give you money. But that wasn't the case for Carter Vallis. They sent me during 3 years $750,000 as royalty due to me, and it is with this money that I made the Talwar Research Foundation to build up a lab where I can still continue to work. You see, as has been told, I was at Medical Institute for 27 years, AIMS. Then I built up from zero the National Institute of Immunology, and thereafter, I didn't know, I was for 5 years at the International Centre for Genetic Engineering and Biotechnology, ICGB. But even after when I was 73, and they cannot, their regulations, they cannot renew after the year, after 70 years. So therefore I didn't know what to do, and with God's grace I got money to start my own lab, where I continue to work even today, though I am 97. So here is the test that we did, that is it indeed an antibody highly specific, the PIPP, and it reacts or does not react with other. On the other hand, you see, what we noticed was that with normal tissues it doesn't react. But with adenocarcinomas, whether it is of lung, of pancreas, bladder, colorectal cancers, hepatocellular carcinoma, and colon cancers, all these 7 types of cancers that we tested with, it had reactivity. So that indicated that all these cancers of advanced nature express HCG, whereas normal organs don't express HCG at all. That is the rationale in choosing HCG as a immunotherapeutic target, so to say, for cancers. Next slide. Here is another proof that indeed our antibody would go only to the tumor. Here is iodinated antibody injected into a mouse where the tumor has been implanted. It goes straight to the tumor and nowhere else. Whereas an irrelevant monoclonal antibody iodinated will spread everywhere in the body. It is not going focused and localized to the point where the tumor is. Therefore, you see, the approach will be highly safe. It will only go to the tumor and not elsewhere. Next slide. Here is another experiment done to determine that you can prevent the human and even cure the human lung cancer cell tumor in nude mouse. You see these tumors, the human tumors, they grow in the nude mouse, as most of you know. And you see on the top, where no antibody is given, the tumor grows. You see, I wonder whether you can see the size of the tumor growing in the C and D. A to B to C and D, it has grown. And then when you give the anti-HCG antibody in increasing concentrations, you come to a point where there is no tumor growing at all here. So it's, in other words, this antibody can prevent and even can cure a tumor which is expressing HCG. This is the lung tumor. Next slide. Next slide. Here is an actual experiment in patients, and in their, you see, in their tumors, from control where there is no binding, you come to concentrations 50, 100, 200, 40, 400, 800, 1600, and control, of course, there will be none. But at 1 to 50 dilutions of this PIPP, this is a very good antibody, highly specific and high affinity antibody. It can bind with 96% of the lung carcinomas. So therefore, you know, this antibody can go straight to the tumor rather than elsewhere in the body. Next slide. Then we tried that, well, in order to use many times, sometimes the antibody itself cannot kill a cell. So why not make a composite with something where the antibody can home it to the site where the tumor is, but it should have another molecule which can do the cytotoxicity. So we linked it up with curcumin. By the way, curcumin is, everybody knows it's haldi, but the essential part there, the chemical part, is curcumin. And what we did was, we linked our antibody, PIPP, with curcumin, which is, PIPP is chimeric, where we have humanized a part of it, from the mouse to humanized form. And if we give curcumin, make a complex with the curcumin conjugated to PIPP, you get this last part you see is the one that you can make in the laboratory. Or next slide. And if this is used in a tumor, the increasing concentrations of the conjugate of the antibody linked to curcumin kills nearly 100% of these tumor cells. So therefore, here is an approach where you can develop a therapy, a new type of therapy for such cancers which are very difficult to treat and which lead eventually and invariably to the death of the individual. So you can cure him with this type of conjugates. Next slide. Next slide, please. This is how it binds to the CIPP and CIPPI curcumin. They both are tested here on mold 4 cells, which are lymphoblastic leukemia cells. And you see only the medium, these are the cells. Then the mold 4 cells with 50 micrograms of curcumin antibody alone, and then with antibody alone, and right below is antibody linked to curcumin. And you see every tumor cell is killed, has melted away. So therefore it's a pretty good conjugate to be used for therapy of such tumors. Next slide. Now we made, you see the curcumin is yellow and fairly yellow, and everybody doesn't like to have that in. So we made another conjugate, and this time it was with sarsen. Sarsen is a toxin, and it's also very good to kill the cells. So again here we should take the humanized single chain of our antibody, PIPP, which can home the conjugate to the point where the tumor is, and sarsen linked to it is the molecule which will act to kill it. Next slide. This is how it is purified and tested. After when you make it, you have to purify it, and the final product you can come to a single dot, which means it is now purified sufficiently into a single entity, the PIPP immunotoxin. Next slide. This is what it does. On pancreatic cells, look how it acts. Again, from that side is 1, and this side is increasing concentration. As you increase, and the 3 colors are the observations at 3 time points, 24 hours, 48 hours, and 72 hours. You see at 1 microgram, micromolar dose, you have small killing, and you keep on increasing at the 10 micromolar concentration, you have almost 90% of cells which are killed in vivo. It's the pancreatic cancers. Next slide. This is the colorectal cancer. Again, the colorectal cancer which expresses, it's the advanced stage expressing ectopically HCG. You can again kill at 10 microgram about 90% of cells. So 90% or beyond. So it's a pretty good killing is done on tumors which are already established. In other words, it's not prevention, it's now a cure type of an approach, experiment to cure it with such entities. Next slide, please. And this is how, you see, all this was being done in vitro, but it's always essential to determine also whether it will work in vivo, because everything that works in vitro may not work in vivo. And this we have done in nude mouse. In nude mouse, you can put in the human tumor, and so colo 205 cancer cells were injected in nude mouse, and then the tumor developed. We let it develop into a tumor. At that time, you do the injection of the immunotoxin, and then you see whether the tumor growth continues or is it arrested, and maybe even reduce the tumor size. Next slide. This is how it looks. In the control, you have this tumor in the nude mouse in vivo, and in the test one, you have a much reduced tumor. So it has acted, and it has sort of reduced the size of the tumor. From this growth, it is by this treatment, you reduce the tumor. Therefore, it is a therapeutic effect on a tumor which is already developed. Next slide. This is another tumor, another test, and the same thing is observed in the control, and with the conjugate tested, there you are. It's much less the tumor. Next slide. Now I'm going, so therefore you see this is about the HCG and HCG expressing tumors, advanced stage tumors which express ectopically HCG. Now I'm going to take you to a cancer which is quite frequent. It is the HPV, the carcinoma of the cervix. In women, it's quite often. The HPV is a virus which causes that cancer. And for that we made a Basant. It's a nice name, isn't it? But Basant is composed of 4 ingredients. Curcumin, which we saw was a good target, but you see, even as such, it makes everything yellow. But given in these ampules, by the way, this has gone right up to the end and gone to industry. It is given to a Kerala company, Bifa Pharma, and they make it and encapsulate it into such type of ampules. They are very easy to handle, and they don't, you can insert it into the vagina very easily while going to bed. And this is what Basant is composed of: curcumin, amla, extracts of neem leaves. You heard about the Ayurveda and aloe vera. So we have these 4 different ingredients, all pretty well known in our tradition to have their effect. The combination of them works much better than any one of them individually. Next slide. It inhibits the growth of Neisseria gonorrhea, which is again an infection to avoid. And even those strains of gonorrhea which are penicillin, tetracycline, and other antibiotic resistant strains, it still is inhibitory with this entity. Then it has inhibitory action on candidas, all types of candidas. So you see, these are the infections which women suffer very largely. You see, at the medical institute, 50 to 60% of women coming to OB and GY outpatient had abnormal vaginal discharge. You know, it was full of discharge and whitish looking vaginas. I have pictures, but I haven't brought them anyway. Now, you know, this basant also helps clear all the candidas of various types in the vagina, in vivo. Next slide. And then, you know, I come to what I wrote was cancer. HPV is something which is a virus which causes the carcinoma of the cervix, and it's not a pleasant thing to have. And our basant inhibits the entry of HPV-16 in HeLa cells, and this is not only determined here but also abroad. Next slide. And the therapeutic action on HPV was determined by a trial conducted in Aligarh University Medical College by Dr. Rajshri Sharma, and the BC Das enabled us to have contact with each other. Therefore, I put his name too. He was an intermediary to put us together. Next slide. Next slide, please. Patients screened for HPV DNA 159. Out of that, 19 were positive for HPV 16, and then with written consent, 11 of them agreed to undergo the trial that we were wanting to do. And next slide, please. And here is the result. This result showed that all of them, 1 to 11, all positive for HPV to begin with pre-treatment. Post-treatment, all of them were negative. No HPV, no PAP. So it's, even to me, it was a very wonderful result. 100% is not always observed, but here it was a 100% effect. On every woman who had this infection, she became negative. And that is how this was ready to go to a company, and PIFA Pharma took it. Unfortunately, these companies, they sell abroad more than what they sell within India, partly because they make much more money. So he sells a lot to USA, but to India, I am told it's not available. It's the same thing about my vaccine for tuberculosis and leprosy, which is Mycobacterium indicus prani, MIP. You see this we developed against leprosy. Also, it's good for tuberculosis, both immunotherapy as well as immunoprophylaxis. It was given to Kerala. And in January, when I attended the meeting of nephrologists and dermatologists, they said, well, can you help us in getting some MIP from Kerala? It doesn't supply us very much. And I asked, why, what was wrong with them? he sells it for psoriasis and for cancers. The same vaccine is a potent invigorator of immunity and has effect on many things, good effect. And he makes money there. These companies, you see, this is the lesson that message I'm passing you, that even good companies are more interested in money than in serving our own patients. Next slide, please. So here is the credits. Jignesh Gupta is my associate. Kirti Nan is the one who is doing current work on she's in our lab, and she's quite talented youngster and is doing good work. Then Kritika Sonar is again one of the members who is currently with us, and she's, as you see, from Bengal. And then these are the Hemant was earlier my student, now God knows he is somewhere in USA, and Vishwakarma and John Schiller, Rajshri Sharma was the obstetrician who did the trial that I presented. B.C. Das is the one who put us together. So therefore, this is all that I have to come to say today in the topic that I gave, that it is possible to have immunotherapy of cancers which are difficult to treat, which are aggressive, and which are drugs resistant, and which express ectopically HCD. Thank you very much.